Sorry about the delay. I'd like to thank the organizer for giving me the opportunity to talk here. So to this audience, there's no need to introduce the role of chromatin in gene regulations, but I just want to emphasize that by looking at the static pattern of the chromatin states alone would not be very informative when you think about the gene regulation, just like what Shirley said in her previous talk. By looking at the peaks at one condition, there's very little information you can get out of that. So what you really have to think about is that the dynamic change of these patterns from the cell type to cell type, because that's the one that's really driving the cell type specific gene regulation. A very well-known example to illustrate that was shown by Bing Ren's group. It doesn't work. So I'll just use my word. So if you see on the top level, so that's the promoter level. So you see basically there's very little change of epigenetic patterns between the cell lines. So that means that if you just look at thank you. If you just look at the peak over here, no matter how strong this peak is, that does not correlate with the gene expression changes. Whereas if you look at enhancers, so you'll get a lot more information. So fortunately, the ENCODE and epigenome roadmaps, they generate tons of the data in the multiple cell lines, and giving us the ability to systematically look at the variability across multiple cell lines. So I'm giving you an example for K27 trimethylation, but you can do this for other marks as well. So by looking at this, each of this track represents a cell line, and this is all for this one single mark. So you can see that for most of these areas shown here, there's really little activities around here, but there's these highlighted regions. You do see these, not just the activity, but also the variation of that. And from that, we can estimate which regions are variable across the cell lines and which ones are not. One easy way to do this, of course, you can say, well, how about just to use the variance to quantify the variability? But actually, that's a very bad choice. And the reason for that is that if you plot the mean versus the variance of the signals, there is a very, very strong correlation that you would expect for the Poisson distribution. So what you really need to do is to take that correlation into account and define a score we call the IOD, index of dispersion. And here we use that to call this plasticity score. And from that, we define two type of regions. One is called the highly plastic regions, which are the regions that have highest scores, and the low plastic regions, with the ones that have the lowest scores. Main focus is on the HPRs, the highly plastic regions. The other one just uses the control. So by defining that, we want to know if these regions we selected are really meaningful. By that, we look at several things. One is to look at conservation. We look at the variation of the DNA methylation. We look at the DNA's hypersensitivity size. We look at this variation of the gene expression. And for all that, it seems that consistent with the concept that these regions selected for regulatory regions. We also do this for the enrichment of these regulatory elements, and you see there's a high enrichment over there. So we want to know what kind of determines this variability. And from that, our first goal is to say, can we predict these regions by using the DNA sequence information alone? And actually, it does pretty good. In fact, that just using the CPG model alone, it does a reasonably good job. The reason that it still does not capture that well by using the full DNA sequence information is that it can only look at these CPG-rich regions, the CPG islands close the HPRs. But if you take other sequence features, it can also give you information about the CPG-poor regions. So, and this is actually a snapshot of that. We can see that the prediction actually captured pretty well with this the variability here, and not just in the CPG-rich regions, but also in the poor regions as well. But it doesn't mean that the signal can be fully predicted by the DNA sequence. As you can see, the correlation overall genome-wide is still relatively low. So there is some correlation, but of course that's not the only story. So we want to go more specifically to see, like in a specific cell line, so what are the mechanisms that regulate the change of the specific patterns in cell lines. And from that, we build a pipeline to find what are the regulators that are mediating those variations. Probably everybody in this room does. What you would do is that you take these regions, select the regions specific in those regions, in each specific cell type, and then we would do a motif enrichment analysis. And the way we did that was to first build a background model to control for the CPG distribution induced bias. And from that, we find that a very good effect to remove the false positive motif enrichment. But that's actually, to us, is still not the end of the world. I mean, one major problem with this type of analysis that, as you all know, that the transcription factor to motif mapping is not unique. And the enrichment of the motif, I mean, for each motif has its own quality. So some of them are more specific, some of them are not specific. Just by using the enrichment analysis alone, it's not going to be that informative. In fact, if you just do this alone, you will find a lot of motifs that are enriched with those regions. So what we extended are to add these two additional steps, and one is to look at the centrality, saying that if there's a really specific regulatory role, then it has to be enriched only in the center of the peaks, but not in the flanking regions. And we also included this gene expression data in the way that if the true factor that's mediating, then the expression level of the transfer factor should be anti-correlated with the expression level of the target genes. So just to give you an example, we tried this on this cell line GM1 to A71. So we find PAX5 is one of the motifs that's enriched in here. And we see there's a very good anti-correlation between the expression level of PAX5 and target genes. And then we see there's a good enrichment in the middle. And we take that to compare with ChIP-seq data in the literature. And we did find there's a really high colocalization between the PAX5 and the K27 trimethylene. In fact, there's also literature studies that PAX5 may have a role in recruiting polycomb, which is the factor that's at the K27 trimethylation mark. So by looking at 19 cell lines in ENCODE, we make up a table with 41 predictions. So this is all public available. If you're interested in any one of these factors, you can look it up. So next we want to say, what if you individual lab, you generate your own data. So in this case, we look at, say, Pro-E. You can do whatever cell lines you like. And then say, can I use the information, the vast information you get from ENCODE to learn things about the specific activity in your cell lines? And the truth is that you can. So basically, you put your cell lines with all the cell lines that's available and selected these Z HPRs, which are the ones that's uniquely active in your cell lines. And in this case, we find that these Tau1 as enriched in cell lines, enriched in this Pro-E. It's anticorrelated. It's highly enriched. And then we did the experimental validation to show that is indeed the case. Interestingly, the Tau-1 factor is known to be a master regulator in this red blood cell development, but only for its active role in the red cell development. So what this suggests is that Tau-1 also have a repressive role in recruiting the polycomb complex to repress the enhancers that are used in other lineages. So if you don't use this kind of analysis, you probably will miss that. So we want to go beyond this and to see, can you do something like with multiple histone marks? Oh, sorry. Before that, I should mention that, by the way, so the computational pipeline is available thanks to our talented postdoc, Luca Pinello. And what it does is actually, it's very simple. So you just provide the band files yourself and use this kind of a simple command, and with the output, it gives you this kind of useful information. And here is a kind of a snapshot of what it would look like. This is your factor. These are the numbers. And these kind of show you the reports that you can get. If you click on each of those, they will give you the detailed output of each individual factors. So it's not online yet, but we hope that it will be soon, in perhaps 1 or 2 months. And hopefully this will be the kind of location we'll put it on. So we want to extend this to multiple histone marks. And as Jason and Manolis beautifully did, so you can detect combinations of multiple histone modification marks by using a beautiful tool called chromatin-state. So using this chromatin-state, what we can look at is the variability of the combinatorial marks across, combinatorial states across the multiple cell lines. Here, this is this in each of the cell lines, and to measure the variability, we use this well-known quantity called the entropy to measure that, and you can see that the entropy kind of find interesting regions. In this particular case, that the enhancers that enriched specific to this cell line, K562 cell lines, and you see the clusters of those. That actually, you probably cannot see that, but it's actually encompasses a number of the GWAS identified trait, SNPs traits, which means that perhaps using this kind of variability analysis, you will be able to find the functional annotations of some of this GWAS variants, with something that we're interested to do as well. This slide is just to show you that what you can also see is that there is a coordinated patterns of the switch between the different states in different cell lines and the different states. You can identify these modules, and by looking at these modules, you may be able to see some kind of themes and functions. So you can also look at the co-variability of the chromatin states and neighboring states, and what this shows you is something kind of interesting. So some of the regions that are really have this kind of block regions, but the regions that lose this correlation, that correspond to the CTCF binding sites consistent with the role of the CTCF insulator. So I hope that I convince you. The take-home message I want to say is that you have to look at the chromatin variation across the cell lines, not just to look at the snapshot itself. And with that, I would just want to acknowledge the people who did the work. The main work is done by a talented postdoc, Luca Pinello. This is a wonderful collaboration with Stu Orkin's lab, in particular Jian Xu. Thank you very much.
We have time for some questions. I'll start one up. Have you run H3K9, 3 through the same pipeline as you did H3K27?
So we have not. So far, we only look at the K27 trimethylation. Yeah. So I would expect that if you do the K27 acetylation, then a lot of regions will overlap with the regions we identified over here. But we haven't done that yet.
by not looking at the varying chromatin-state region. So instead, we're focusing on the dynamic. You want to focus on the HDRs, right? And
Yes.
And look for enrichment peaks in there and to identify PFs that might
Yes.
be relevant. But it's entirely possible for several PFs to work off generally open regions, generally active enhancers. Doesn't have to be varying enhancers, right?
Right.
How much of the signal? What's the sensitivity like in terms of identifying the PF?
So, if I understand you correctly, you're asking if are there any other transcription factors who are not targeting these regions? So are we able to capture that? Right. Right. So I hope that that's kind of the question that we can only address by looking at the full chromatin states variability, maybe looking at each state individually and so on. But my suspicion is that even by looking at things like individual marker like K27 trimethylation, then you will be able to see a lot of them. And the reason I'm saying that is that the people have look at these things in enhancer changes, this and that, and it's not just mediated by a single mark. That's for sure. It's not just by K27 acylation or K41 methylation. It is mediated by many coordinated work of many other marks as well. So my thinking is that the activity changes of this may be coordinated with the changes of the enhancer specific marks. So therefore, you will still be able to capture a lot of those factors as well. We can discuss later. I think I'm late already.
We should move on to the next speaker.